Hello, everybody. Welcome to the Kandilli Deep Talks seminar series hosted by the Boğaziçi University Center for Life Sciences in Istanbul. My name is Pascal Kalem, and I'm delighted to chair this second webinar of the series with distinguished Professor Utkan Demirci as keynote speaker. Professor Demirci is interim director and division chief at the Canary Center for Cancer Early Detection in the Department of Radiology at Stanford University School of Medicine in the USA. The webinar will start with a presentation of our Life Science Center by Professor Cengiz-Han Öztürk. I will then present the technical assistance project that is funding this webinar, a project co-funded by the European Union and the Republic of Turkey. The webinar will end with a session of question and answers. And if you want to ask a question during the webinar, please write it on the chat of the Zoom platform or raise the hand using the tool at the bottom in the menu. Please note that this webinar is recorded. And I would like to thank the technical team for preparing the webinar and providing simultaneous translation. And actually, you can select the audio channel on the Zoom platform using the menu at the bottom, choosing interpretation and English or Turkish. So I will now leave the floor to Professor Cengiz-Han Öztürk to present the Life Science Center. Professor Öztürk is the Industrial Projects Coordinator at the Boğaziçi University Center for Life Sciences and Technologies. I will now stop sharing my screen and I give you the floor, Professor Öztürk.
Sorry, I'm having a little bit technical problems here, but I guess I will speak Turkish and there will be simultaneous translation. That was the norm last time, so I switched to Turkish now.
Okay.
Ben de herkese hoş geldiniz diyorum. Özellikle Pascal'a teşekkür ederim.
Welcome everyone. Thank you, Pascal and System Global team, and for all the supporting team. I would like to thank them. Without further ado, I will be repeating a short presentation mentioning and explaining the center, and I will be sharing some updates with you. Of course, I would like to give my thanks to Utkan. I am looking forward to his presentation. After this short introduction, I'll be giving the floor to him. Okay, I'm trying to share my screen. Is it full screen now? I think it is. Was I able to share my screen? Yes. Okay. So regarding Life Sciences Technology Research Center, I especially supported the establishment of the center, and also I am now supporting the Kandilli part of the project. So I would like to introduce you Kandilli campus. This deep technology base is not affiliated to any academic unit. So Uygar Center established a foundation. It is being supported by Life Sciences and Technologies Uygar Center. And also in this building, we do have Biomedical Engineering Institute, Chemistry, Physics, Chemistry, Engineering. We do have labs of academicians coming from these departments. We do have conference rooms, meeting rooms, classrooms in the building, and also within the campus. We are physically close to some other buildings. And regarding raising awareness about deep technology, Before this EPA project, we started our activities, but with some various activities that we are having at the moment, we are accelerating the promotion of our center. So we are like going one step beyond. So there is an interesting realization design of the center. So there are academicians or students. There were academicians and students coming from Boğaziçi and out of Boğaziçi. So it was an incubation activity. So there's an incubation activity here. So on the opposite of that, there is the Life Sciences building. So from this EPA project, some firms are being provided technical assistance to use this infrastructure, and their coordination is being increased. And also some new infrastructures are established in our center. In our center, we are hosting more than 50 academicians. So it's been 10 years now that we have established the center. So it is an academic-centered center. It has many academic studies. And in terms of technology production or targeted treatment technologies, it will be providing services to the industry, and it will create added value in the sector. So it has such a strategic objective. So here you see the issues that we are working on: vaccine, intrabody microsystems, smart devices, smart medication transfer systems. So we are coming around such a theme. There are more than 200 students, masters and PhD students being supported by the center. An important group of these academicians are working within the team of targeted treatment. And we are aiming to provide infrastructure to all researchers. And also we are trying to create regional economic contribution. And in terms of its administration from our cluster project, with some cooperation and protocols, an important team contributing to the infrastructure and also entrepreneurship activities of the center. And they are also actively involved in the execution of the SIPA projects. Here you see how the center evolved in the various presentations before I mentioned this. The important thing here is that in operational terms, there are some strategic plans, so they are updated, and for the future, some new important project assistances were provided. So it has submitted these proposals, but we are focusing on targeted treatment. We do have a vivarium, test analysis unit, clean room. Our vivarium is accredited. ALAC International is leader, and it is also in Turkey. We have updated it. This is our clean room. So it is dealing with smart devices, and it is also certified. And this is the test analysis unit, which is being used by many researchers. So chemical analyses, physical characterizations are going on here. And some of them are subject to some kinds of accreditation, and they will be providing services in the new building. We do have two projects here. One is this, so project number one, and project number second is being supported by the Competitive Sectors Program. It is a project named Independent, about biorefineries. So this technical assistance package is mostly contributing to the infrastructure of the project number one and supporting SMEs within the scope of the project one. But in terms of the sustainability of the institutional structure, in terms of SMEs benefiting, and in terms of sectoral studies and feasibility studies, project number two, Independent, is importantly linked with our project. So we are also supporting that project in terms of Kandilli campus activities, and also in Sarıyer, where Independent project is located in Sarıtepe. So we are also touching their activities. In this project, we have envisaged these to happen. So we'll be buying some machines, so we'll be having some production facilities, and we'll be strengthening the material characterization lab. We can show some things here because we have, so here you see the developments under independent project here. And this is about its development of sectoral pillar. And in terms of sustainable cooperation, this technical assistance package is also very important for us. So we will be touching upon these issues too. And within the entrepreneurship ecosystem, to reach the right people in Kandilli campus, we are doing some important deeds, and also we are strategically cooperating with Technopark Istanbul, with which we do have a protocol. So we are developing the system. On the other hand, we are also realizing, designing this infrastructure that this ecosystem will support. And Innovita platform is the interface of us, the coordination team. And Istanbul Health Industry Cluster is also involved. So we do have two separate projects. One is Kandilli Health, using these infrastructures, and the other one is BioCube in Technopark Istanbul. These are being supported by regional development agencies. I'll be showing some updates. So this is from the plan. In every presentation, we are evolving more. Now we have the infrastructure. We do have the first admissions. Now the contracts are about to be ready. As of June, first BioCube entrepreneurs will be in. And in Kandilli Health Project program, the first trainings were carried out, and screening or imaging unit is accredited and training started. Devices are accepted. I was going to share some photos about that, but I had a technical problem, so you can follow our social media account. And STEM device is also commissioned. Now it is providing services in a very wide scope, especially environmental STEM and biomaterials. It will be providing specific contribution to some certain types of studies. It may replace some modules. So this might be something new. You might have seen on news regarding Boğaziçi University, and between an investment club, there was a cooperation agreement. And within bonevest.com, these ecosystem entrepreneurs in academic terms, on the other hand, of course, in terms of the outer entrepreneurs who would like to use our infrastructure will be supported. So on the 9th of May, this has been signed. So it's a very new start, but I think there will be a quick development. There is a comprehensive founding board, but they will be working as a coordination team, and many investment networks, individually and institutionally, will be called for. So this network will expand, and it will be strengthened by the network of Boğaziçi University. And of course, we're focusing on health sciences here, but it will be expanding to the other fields too. So such an investment network will be also supporting the entrepreneurs which are making use of the infrastructures of this IPA project. And as entrepreneurs get stronger, our infrastructure will be more sustainable. Now I will stop here. I wanted to share some developments with you. Because of a technical glitch, I couldn't present the presentation that I wanted to, but I think gave you some general opinion. I know you haven't come to listen to me. The keynote speaker is Utkan, so he always has very interesting presentations. I am looking forward to listen to his presentation too. So we'll see what he is doing. So what kinds of developments happened recently. So he'll be talking about these. Thank you very much for your attention.
Thank you very much, Professor Öztürk. I will now start presenting the project that is funding this webinar. I had a little glitch in my computer. I don't know, it's because of the switch of screens. So I'm going to share again my screen here. Okay. I'm going to ask the assistants to tell me if they can see my screen in presentation mode. Could you please tell me if you can see my screen in presentation mode? Thank you. Okay, so I will provide now an overview of the technical assistance project that is funding this webinar. I am the project team leader of the project, and I'm located at the Life Sciences Center, which is at the Kandilli campus of the Boğaziçi University. This technical assistance project is co-financed by the European Union and the Republic of Turkey under the Competitive Sectors Program executed by the Ministry of Industry and Technology. In this short presentation, I will present the project very quickly, its activities, and the opportunities that the project is providing to SMEs and also for the center itself. So this funding by the European Commission is part of these instruments for pre-accession assistance called IPA, and is the means by which EU supports reforms in the enlargement countries with financial and technical help. So the IPA funds build up the capacities of the countries throughout the accession process, resulting in progressive positive developments in the region. The overall objective of the project is to accelerate the research and the development activities of SMEs active in the life sciences domain, with a focus on specific cutting-edge technologies and high value-added products. So I'm showing the picture here of one of the sites where these services are operated, but as presented previously by Professor Öztürk, there are several sites around Istanbul that provide these services of the Life Science Center. So the purpose of this particular project is to provide support to the Life Science Center, to augment the current technical capabilities of the center, and to add a new infrastructure to the Life Science Center, providing links to the regional industry with a focus to improve its cooperation with regional industrial partners. More specifically, to expand its support services regarding preclinical animal imaging experiments and specific material characterization tests to the local SMEs involved with specific research and development activities related with in vivo medical devices, or biotechnological products, and biomaterials. The parties are involved in this project are the contracting authority, that is the Ministry of Industry and Technology, and the Directorate of EU Financial Programs. The second one is the European Union delegation, the EUD. There is the Boğaziçi University, which is the end recipient of the assistance, and the contractor that is implementing the project itself, that is the company System Global. The project started on the 1st November 2021 with a duration of 18 months, and the base of operation is Istanbul. So in this slide, I'm showing you the services that are already existing in the Life Science Center and the services that are going to be added to the Life Science Center with the support of this European and Turkish project. So the existing services are the clean room, the vivarium, the test and analysis. As presented before, these are operational, and as you can see, they are really high-tech services. Now, this project is supporting the development and the implementation of new services, specifically the small animal imaging services with imaging technologies such as MR, PET, and CT. Pilot production is also a new service that is going to be proposed, and microalgae bioproduction. And these services will be all available and running at the end of this project that is planned in about less than a year. So the main objective of these developments is to provide support to startups and small SMEs in the domains of pharmaceutical industry, medical devices, and biotechnology. So just a quick overview on the activities of this project. The activities are divided in two types. A type that we call A, which is directed to the support to the SMEs and the industrial potential clients of the center. And for these, an analysis of the market has been conducted in the last months and is going to be presented to the SMEs. The markets that has been studied are the markets of interest for these, which are pharmaceutical industry, medical devices manufacturing, and biotechnology. These analyses are going to be presented to SMEs directly through focus group meetings, where SMEs are going to be selected to receive this information. A selection of SMEs are going to also receive for free what we call SME boost support, which are composed of training in terms of accessing the services, but also in terms of training about development of SMEs and business development. And they will also receive consulting support in law, in patents, etc. And the second set of activities are the activities B that are related to the development of the Life Science Center itself, with the review of the Life Science Center operations, the establishment of business development unit and a business development plan to manage the old and the new services that are going to be provided. And of course, there is a support to communication and visibility activities. And overall, I insist again, it's by all these activities, we intend to support startups and small SMEs in the three sectors that I mentioned before. So opportunities that this project provides are at the level of technical services, because the new services that are going to be installed, small animal imaging, pilot production, are services that are unique in Turkey. And in particular, these machines, this equipment for small animal imaging. And therefore, we are very happy to provide these new services to the community of SMEs and life science in general. On the other hand, we have business-related services that are funded by this project. As I mentioned before, we are going to provide individual diagnostic analysis of 60 selected SMEs by July 2022. The SMEs have already been contacted and they are selected according to their profiles and their areas of activities. We are going to run focus group meetings also, presenting market analysis results with gaps, opportunities that we have been detecting both at the national level, but also at the global level. And last but not least, SMEs will be able to apply for participating in this boost support that will provide these services, training and consulting services. And we have already started, for example, these talks, Kandilli Deep Talks, are part of this support that we are providing for free. Focused thematic trainings are going to be provided and individual boost support. So stay tuned, because there are going to be some calls that will be announced in the next months to be able to apply for these services. So with this, I just want to acknowledge the teams that are participating to this project. There's the technical assistance team that is led by myself and Professor Emir Baki Denbash. And we have a list of non-key experts that we have recruited specifically for this project. There are national non-key experts and also international ones. I want to acknowledge also the team of System Global with the backstopping team that is supporting the administration of the project and the management. Also the partners that are ICADA and Congenia. And also, of course, the Life Sciences Center in general with all these members, the Boğaziçi University, the Ministry of Industry and Technology, and the EU delegation. So I have finished my presentation of the project, and I'm going now to introduce the next talk from our keynote speaker, Professor Utkan Demirci. And before giving him the word, I would like to present him a little bit to you. So Professor Utkan Demirci is a professor with tenure at Stanford University School of Medicine, and he serves as interim director and division chief at the Canary Center for Cancer Early Detection in the Department of Radiology. He also was a faculty member earlier at the Brigham and Women's Hospital, Harvard Medical School, at the Harvard MIT Health Sciences and Technology Division prior to his appointment at Stanford. Professor Demirci's group is focused on the development of point-of-care technologies and creating microfluidic platforms for sorting rare cells and exosomes for infectious diseases and cancer. He received his PhD from Stanford University in electrical engineering in 2005, and he has published now over 200 peer-reviewed articles. Professor Demirci's seminal work in microfluidics has led to the development of innovative platform technologies in medicine, and his inventions have been licensed to numerous companies. He has co-founded several startups and serves as advisor to multiple companies. So Professor Demirci, welcome, and we are eager to hear your presentation. So I will stop my sharing my screen now, and you are welcome to share your screen.
Thank you very much. I thank you and Cengiz Hocam and Emir Baki Hocam for the opportunity to be with you today. And you know, it's a pleasure always to contribute and work with Boğaziçi and the team here, especially in this outstanding center. So I want to kind of walk you today through different lifetime, almost like a decade long lifetime of one of the technologies that we have developed, created, patented, translated. I started a company on it. There are other things that I wanted to also could go through at different levels of life cycles, as I've started quite a few companies in US as well as in Turkey. But I just wanted to kind of walk us through today one of the life cycle from a technology that's being invented to finding its niche as a tool into the actual clinical practice and still continuing its lifetime in the hands of the clinicians as they use it more, discover more things, and they are the real judges of how it really impacts their clinical practice. So before I go there, as you noted, I'm at Kandilli Center at Stanford. I'm currently the interim director of this center, and we have people from all different backgrounds in this center among core faculty and beyond, trying to develop on our side tools and technologies, but it has got like the center that you are describing, the strength of it is people with very different backgrounds coming together and then converging around a common goal, a common mission to develop new early detection technologies for diseases, especially with the focus around cancer. Switching over to some of the work that we have done, I have had a engineering background over the years that kind of culminated and transformed into more like a biology and medicine type of applications, kind of in a melting pot, and it has led to different stages of development. But something that I always see in the development of these different tools and technologies that we come up with, it always starts with a real need that we identify either technologically or with the help of a clinician that's out there in the clinic. And then we either develop a new innovative tool ourselves, or find and learn things and develop the next generation of things to solve a problem. And then it becomes a new direction into the lifetime of this problem, where it takes sometimes years, sometimes decades to kind of really develop something worthwhile. And I actually kind of really totally agree with this phrase where it goes, simplicity is the ultimate sophistication, because really being able to develop something simple in this process really brings you all the way to the end much faster, much easier, with less hiccups going through the challenges, both on the technology side, on the scientific side, as well as on the market and product development side. As you are going through these cycles, if the tool is cleverly simple, then it really actually is a strength. And that's something that we have watched out over the years for the tools that we try to develop. I want to tell you about one specific tool that actually has become applied in the infertility space. Infertility is a huge field where you have more than 70 million couples suffering with infertility globally. But the way we started looking things is with an engineer's perspective, and looking at devices and taking some notes from other inspiring scientists, where I really like the note that says that there is really a lot of space at the bottom. And you might have seen these robotics movies from 60s, where you could actually send these small micromotors into a circulation and address disease and so forth. So when you start looking at the stark difference between scale, comparing an elephant and flea, we start seeing that with scale, everything changes. The physics changes, the hydrodynamics changes, and it becomes more and more interesting to study the micro scale, and even further going down to nanoscale, where your parameters and design parameters, looking at it basically from an engineering side, is significantly changing. So some of the phenomenon that happens in mesoscale doesn't happen the same way as we know in self-assembly. So when one starts looking at sperm, which is the fastest cell in the human body, and starts looking at it almost like a micromotor, you kind of realize that where the tail gets into the head, there is really these nanomotors. There are many mitochondria that pumps a lot of energy, a lot of ATP into the system, that allows the tail of the sperm to make a sinusoidal and cork movement, depending on what stage it's in its movement. But the flagellal movement of sperm has been very interesting. So we started studying this, trying to understand what it means and under what conditions, how sperm actually swims. And that became later on very important, as I'll tell you. But taking a step back and looking at the sperm as a biological cell, over the past decade, sperm has been gaining a lot of attention, especially with the epigenetic and genetic patterns of sperm quality impacting embryo quality in IVF, in ICSI, and in in vitro fertilization. The importance of sperm has been gaining a lot of interest, and sperm comes in all forms and shapes. You can see the double-headed and double-tailed ones, the large globospermic sperm, or pinheads that are really small. And these are easy to identify under a microscope. What we cannot identify under a microscope is, without staining the sperm, is the integrity, or the DNA integrity, or the epigenetics of sperm. And those will become more and more important, especially when we look at environmental factors such as pollution. They all have an impact on the fertility or infertility, and you kind of see that they create DNA fragmentation and DNA damage. And sperm goes through a spermogenesis process that actually differentiates stem cells into sperm, and that whole process becomes very important, especially with the surrounding factors in terms of the selection of sperm. And things like ibuprofen could actually have an impact on one's on sperm. And When you look at the nature and connect a sperm's journey, so to speak, it takes about 5 to 7 days for sperm to follow the walls of the uterus and later on to make it all the way to the egg. And that's a 5 to 7 day journey, which is like a marathon. And only they go through these very little channels, and they start with millions, and only a couple hundred make it there, and only 1 or 2 can actually fertilize the egg, depending on the situation. When you look closer in the mammalians, we see these microgrooves that are channels on the wall that sperm actually follows and follows those trajectories, and which becomes even more interesting in terms of trying to understand the long journey of sperm. So, kind of putting these different pieces of the puzzle, kind of giving you backgrounds, but one thing to look at also on the clinical side now for couples with infertility is the solutions that we have to infertile couples. One of them is AI, artificial insemination, which is referred to as aşılama in Turkey, which is an inexpensive method, but you still have to be able to select the sperm or wash the sperm with a large centrifuge and really lab equipment. When you look at ICSI, which is the most common process, where you take a single egg and you take a single sperm, and then with the needle you create the embryo, and here you only have maybe 10 eggs and many sperm to select from. So which sperm to select for these few eggs so that they can become successful embryos? It becomes an interesting question of cell selection or cell sorting. And the way we do it today, or until these tools that we developed became clinical, we were just centrifuging it, and then letting, after this centrifuge is known to create reactive oxygen species and DNA fragmentation. But on top of that, you're letting the sperm swim up and put them under a microscope, and embryologist with a trained eye picks the most good looking one. And when you look at sperm under a microscope or in a droplet, this is how they look. Some of them are swimming straight, some of them are like turning in circles. The question I asked to many embryologists is, how do you pick which one to pick? And they were like, oh, we try to pick the ones that can keep a straight trajectory and that tend to move forward. So this is a sperm behavior on the left inside the droplet. But when you take sperm and then you start reducing the height of that droplet by using microfluidic channels, one starts seeing really interesting phenomenon. So this is when we start looking at sperm as a micromotor with a sinusoidal wave structure. And when you actually, as I described, reduce that size of the top channel, change the hydrodynamics, make it comparable to the size of a sperm, you start seeing that, and this is done in collaboration with Dr. Tuzel, who was at WPI. We created these models where sperm is actually interacting with momentum transfer with its surrounding fluids. And then those fluids are actually interacting with other sperm movement. And in these constrained environments, sperms start talking to each other hydrodynamically. If you are in a certain specific hydrodynamic region, like in the small microchannels that I have been showing you both in nature and also in the devices that we designed, they start synchronizing with each other. So they are not just swimming randomly. When you look at mice, actually mice have different morphology than human. They have hook heads, and you kind of see that multiple hook head mice sperm, they combine together with these hooks, and then they go, like similar to this video I'm showing to you, they go in sync towards this big race that they are in, so to speak. But we don't have in humans such a behavior where sperm are getting hooked, but apparently they talk to each other under these hydrodynamic conditions. So what we did is we explored this further, where we created periodic arrays of pillars, like the Roman pillars of Ephesus or many things you can find, and these pillars we periodically controlled, and that creates a certain hydrodynamic environment where we are putting sperm into this structure where we can control the sperm's interaction with the pillars. Like if the sperm is a large head, it can get lost, it can kind of hydrodynamically not find its way. But if the sperm has the right morphology and the right engine and the right energy production, you kind of see that it does either of these movements. One it can get almost diverted with the pillars because it is out of sync or out of resonance with the periodic structure, almost light hitting a lake surface and bouncing in or bouncing back. Or it can actually even resonate with the hydrodynamic periodic structure in such a way that it can actually even pick up speed. So you can see sperm in these structures going faster to the outlet. So this whole design turns into a device where we are able to load sperm into a device and then just do a single load step, no centrifuge, no manual process, let them swim and race with each other in an incubator, and half an hour later come back and collect the best sperm at the end, the real swimmers. So this idea actually also kind of correlated, as I'll show you later, with good swimmers that make it to the end of the channel, also being able to have the very low DNA fragmentation. And this is kind of, again, a simulation of multiple sperm. We are tracking 3 of them. The red one is a pinhead, meaning small head sperm. The blue one is a globospermic one, meaning a large head. And the sperm that you see over here that's rapidly going is much faster than all, like many others that are in good shape. And they are likely to synchronize with this structure and then make it to the outlet, tens of minutes before any of these other ones can actually make it. And that's a self-sorting mechanism. These tools became CE approved, and they have been used heavily in Europe, and they've been FDA approved since 2018, and now, last time I checked, it was in 35 states in the US and 245 clinics. It's also in Japan and many other places that I do not track anymore. And especially during COVID, it became so clear that these embryology labs or andrology labs needed standardization, and people could spend so much time at the clinic. So having something standard and with minimum manual factors became more and more critical, and it kind of became an actually accelerating factor for these tools to be adapted over centrifuge. I mean, you kind of can see in semiconductors, we have the faster, cheaper, and so forth analogy. And this kind of actually fits the faster, more efficient and standardization aspect. I'll show you some of the early studies in the hands of the clinicians that kind of made this interesting. And there are like so many papers that are coming out now that I'm not able to track all of them, but I see some of the interesting ones as I gaze through it every ASRM or ESHRE, when a lot of the clinicians that are using it now are publishing their results with the tools, which is really exciting to watch. And this was one of the early studies of its kind. Dr. Rosen, who runs the UCSF Infertility, the IVF clinic, he did a split study with sperm. He tested the density gradient, which is the most expensive method in terms of motility. And we could see that, our motilities were significantly higher, that were sorted out with the microfluidic chip. But what was more exciting in this, on average in human population, you get 10 to 30% DNA fragmentation. Anything beyond 30%, especially 50%, is regarded as a factor for infertility. And fragmented DNA in sperm is not considered to be the best thing, although its impact directly on embryo is broadly discussed in the literature. What we could show was by just letting these cells swim through this device, we could really reduce the DNA fragmentation significantly in this really high, even up to 30% DFI, DNA fragmented chips. I saw, some of the early papers came out of Turkey. The reason for that is with the CE approval, I built the manufacturing side of the devices in Izmir, in the Izmir trade zone or trade-free zone, and also in 9 Eylül, and that's made the chips really readily available with the CE approval for Turkey and Europe. And a lot of the clinics that used it, this was one of them, and there are many other studies that kind of looked at the grade 1 or the top grade, or grade A as they call it, embryos, seeing that there was a positive effect on the quality of the embryos. The one that turned out to be most interesting and has kind of changed a little bit how OBGYN. I'm not talking about the big IVF clinics, but I'm talking about these small polyclinics or small offices in the rural areas of OBGYNs, meaning kadın doğum uzmanları, who actually see patients. With this tool, we were able to enable them to be able to do aşılama or IUI in their setting. And this is a process, as I described to you earlier, is significantly, maybe 10 to 50 times cheaper than going through an IVF process. It's what you need is to be able to kind of pretty much wash the sperm in the clinical setting in this really small town. I don't know, like in Izmir, Ödemiş, for instance, that was one of the early people who adapted this technology with a very small incubator without having to send the sperm to be washed out, by a bus trip, let's say, for multiple hours to a central laboratory to get it washed. You can just do it, standardize it in the office. And this was done in a large study that kind of showed that by standardizing it, there was a significant improvement in the IUI. And, I would say that the success of IUI is on average like 40 to 50% more, depending on some clinical parameters. But if you are hitting like 15% as with IUI, you really actually are really impacting socioeconomically the people from small rural areas and villages, because that means for them not having to spend so much money or not to tap into their savings, or if they don't have savings, then they would have to actually sell what they have, their land, their houses, and whatever, to kind of be able to go to the big city and get treated. And this is having a significant socioeconomic impact. So to further enable the doctors who are in the periphery, who actually want to know if it is a male factor infertility, meaning how could we really quickly check, in a very small peripheral area, if the male had sperm or had enough motility so that IUI would be effective, or it was a really a case where it really needs ICSI. We developed this tool that you can download from the iTunes store, and it's a device that's actually able to take a drop of semen at the comfort of your home or at the office, and without any processing, it kind of generates this video and it kind of uploads it for an analysis. So you could actually do a CASA sperm analyses, which one would have to buy a $15,000 to $30,000 machine and chips to process it by the comfort of your office with a simple disposable device that can do it, which enables further this whole socioeconomic picture I described. So this is a place where the need, we developed the technology. The technology created a really niche market that's really beneficial to the public, and then that actually came with its needs and demands, and we developed a follow-up technology to kind of satisfy that need, to make it affordable and broadly available, almost democratizing the whole IVF process on the IUI side. So just to kind of wrap this up, we kind of know by the trained eyes of an embryologist, we can look at motility, morphology, or with really high magnification microscopes, we can see nuclear compactness by looking at the sperm's head. But what we cannot see without actually staining the sperm, but if you stain the sperm, you can't use it. You can't pick sperm for today with low reactive oxygen species, low DNA fragmentation. And this is what this chip does, ineffectively or indirectly, by letting the sperm self-source itself, almost like they do with their swimming behavior. We kind of are able to select also these additional parameters on top of the others that you can see under a microscope. And one thing I've seen recently, and this is really exciting, some papers are claiming that we are also getting lower EPOD rates in embryos, meaning better chromosome quality embryos. But I think a lot of these things will require thousands of patients of randomized trials. And at the end of the day, I think the real value besides ROS and DNA is going to be standardizing the process and making it broadly available. And so, I kind of walk you through the technology where we first started working around on this and publishing the early papers, 2011, to CE and FDA approvals, and then now making it globally available to all clinics. You start kind of seeing these interesting articles when it becomes commonplace. I saw this in Vogue magazine, which is a fashion magazine, where a Californian doctor from LA region, Kelly Bayek, was saying, these devices, they are named Zymot in Europe, and in US, they are named Fertile and Fertile Plus in Turkey. It's the same device with different names. She was calling it a huge game changer for her clinic, for her practice, which was great to see. And another thing that came out, as I go forward, I just want to acknowledge all these clinicians. Dr. George Mutter was one of the first andrologists I met when I was at the Brigham and Women's Hospital system. And George was great. He had like wonderful questions that allowed us to take the right steps, from early 2010s till today. And then Paul and Mitch has been really great clinicians that I should credit here, giving us a lot of the problem and significant feedback on the devices we developed. And I saw another article on the Scientific American. This wasn't about our tools or our devices, but this was a general fertility is a growing problem in the world. And they picked one picture showing petri dishes and syringes and so forth. And these three devices, which are the tools at the time that we had, now they look a little bit different, was considered as the standard IVF tools that are being used at the IVF clinic, which is great to see, because the whole idea is to replace an existing archaic centrifuge technology with a better one. Now I guess the future steps, in the lifetime of this tool is, to kind of see its positive impact, and more clinical studies will come, and it's going to create its own direction. So from the lab, through the development, through commercialization, and all the way to the clinic, this is like almost like an 11-year life journey of one of the early technologies that we developed. Later on, we built other things and other technologies that are at different stages in their career, so to speak, as technologies. But I believe, going back to this slide, maybe in this life cycle, not necessarily all technologies will make it to the end, and there will be competition and other things. But I guess to start really addressing the problem and seeing the clinical problem and driving from that a solution really helps to kind of go through the whole cycle. This is something that we also do along the way that is really important, to be able to mentor the next generation of scientists, to be able to share what we develop both with clinicians and globally, which makes it more fun and more impactful beyond what we have done and academically in terms of doing science and research, which is where we start, but then it grows into a larger tree. And I would like to thank you for all the presentations. Thank my lab and its members that enjoy what they do and make it more enjoyable for me to go to work every day. And I thank you very much for the opportunity again. I thank Cengiz and Hocam and others such as Emir Baki Hocam, who have let me know and invited me for this opportunity. And thanks for listening, and happy to answer any questions if there are any.
Thank you very much, Professor Demirci, for the kind presentation. It has been fantastic. Unfortunately, we cannot hear the applauses, but I'm sure there would be. I would have one question, if I may start. So if we put your presentation in the context, sorry, if we put back into the context of medicine, could you tell us what is the rate of male-related infertility, and what is the sort of market that you're targeting with your device?
Sure. Yeah, I think it is considered that 30 to 50% of infertility is due to male infertility. Traditionally, what's really interesting in the field is people have studied embryology solutions for culturing the embryos. They've studied egg quite a bit, but sperm has been less considered because there's so many sperm. And there's this thought that when sperm gets into the egg through the acrosome reaction, sperm is cleared out of all its methylations, and doesn't matter as long as it has the genetic material. Egg is forgiving, and egg is gonna be able to repair the sperm. I think, you know, some of the early papers I showed that's showing the epigenetics and certain methylation patterns of sperm having an impact on the embryo quality. There was a Nature paper by Hamoud. I think that kind of started really changing the ball game in terms of what sperm might mean, not only for the embryo quality, also beyond what kind of diseases or diseases are being actually transferred to the embryo and to the born baby that might appear, or they make them more vulnerable to diseases in a later age, such as, you know, diabetes, such as cardiac disease. I think this whole concept is a mine of decades of research that will come out of it. And the more we learn and understand about sperm's role, not about male infertility, but sperm's role in this journey, the more I think it will become important how we select and which sperm we select. If you look at the millions and millions years of evolution, so to speak, the reproductive tract is always preserved in all mammalians. So there's a reason that it is designed such way that the sperm has to swim days among millions to get to the egg. And we just only show a glimpse of it, showing that those good swimmers are actually also good with their DNA fragmentation, which I find very interesting. So okay. I hope I answered your question.
Yes. I would be also worried about the fact that you are somehow selecting also the best swimmers that might also have that might not be always the best. So you might want to have you made comparison between efficiency of the best swimmers and the middle type swimmers in terms of results in the sense of the embryos, quality of embryos, and do you have enough statistics to provide at this level?
Sure, people have been studying and they have a definition of what the good sperm or best sperm should look like, and it's based on studies in I think 70s or 80s. People had done a clinical study and they showed that when they use motile or best motile sperm, that led to better results with the IVF. That's a really old study, and since then, people have looked at different ways of characterizing sperm, morphological characterization, swimming characteristics, all those stuff. And a lot of the stuff I showed you on the tables is actually what we characterize, or what the field of andrology characterizes sperm as when they are picking it. What it cannot characterize it for is DNA fragmentation and reactive oxygen species, because the moment you stain the sperm for those, that sperm you cannot use. So what we are providing is that we are really, by letting them self-sort, picking all those parameters or satisfying them, and on top of it, providing additional parameters that what the field cannot choose for today. So that's the kind of what you're asking is exactly the baseline, and then the standard comparison that we on top of it come and add a couple more advantageous parameters in terms of picking the cell. But beyond that, it also adds a lot of facilitation for the clinical workflow. I think that's where people have found a lot of value. And then you had a market question. So for the IVF, we always thought we would serve the IVF and the clinics, but then it created a completely new IUI market that got enabled, which is, I think, very interesting, because now you are enabling the little town OBGYNs also to be able to do IUI, which is really good for everybody, because if you can get pregnant at your local, polyclinic with your local OBGYN, then you don't have to pay the tens of thousands of whatever dollars to go through ICSI. So it's good for the patients. It's also good for the local economy, I would say. And it's also really good in terms of being able to spread something simple by a simple technology.
Okay, thank you.
Thank
Is there
you.
any other question for Professor Demirci?
May I ask a question?
Sure, please.
I didn't know how to raise the hand. I don't know. I'm still learning this Zoom after 3 years, I guess. Maybe I can even try to ask Turkish. So invite other people to ask questions in either languages, I guess, is allowed. This is very nice, by the way, that the interpretation is doing a great job as far as I can say. This is great. Utkan Hocam, bu yıkıcı teknoloji sayılır diye düşünüyorum.
Dear Utkan, we can name it as destructive technology. So clinics. So increasing success rates, is it in favor of clinics all the time or not? And also the public sector, I think, should incentivize this or promote this somehow. So these applications, so public payments, for example, are they standardized in some countries? So how is the situation in the States? I can first answer your first question. So we can impact on petri dish side mostly, because the healing of embryos, so there maybe we can create an impact, then it would be good. So there are some people telling this, but in terms of increasing pregnancy rates, it covers a 9-month period, controlling a 9-month period. So it is not possible with the sperm selection in the beginning. Increasing pregnancy rate can be a very remote goal. There are many factors included indeed, but in the beginning, selecting a quality embryo, for example, you do have 10 eggs, if you will come out with 14 embryos, if you can make it 5 or 2, or the chromosome quality can be higher, then another surgical process, another egg collection process will be abolished, will be eliminated, then this is an advantage in financial terms and also in terms of decreasing the medical processes. This is an advantage. So the coming technologies, advancing technologies might be destructive, but it is destructive for centrifuge. So it is facilitating life of embryo. It is facilitating the flow of the clinic, especially if volumes are very high. During the pandemic, for example, we didn't have time, but we had higher volumes. So any efficient or efficiency increasing technology can be an advantage for ART, especially in the States. In the States, it was so. COVID-19 pandemic closed clinics and it limited the access of the clinics. So it happened that we could just be 2 in a lab, or we needed to take shifts. So the embryology clinics were also impacted the same way. For example, in the embryology clinics, you can say this, for example, I've put the sperm and it is there. So it can have contributions in practical terms. And it can be effective contributions also it may have, especially at the very first step in terms of its dissemination, it can work. Then public authorities, have any of them defined this as golden standard? Is there such a tendency? I think this is not something required or wanted, because public authorities, both in the States here or in Europe, when you have a business perspective, because they are giving very low prices, then China, because this is not a China-like big market, so you cannot, of course, earn from just selling. And compensation by public authorities or incentivizing. So many experts name this as a disadvantage, especially in Europe. I don't know Turkey, but public sector, for example, when it prices something, you cannot have the chance to sell for a higher price. For example, if you do produce it for $10, if someone sells it for $11, then it cannot be produced again because the equation doesn't work for the workers or for the manufacturers, employees. But public incentives for a certain part can be an advantage because in a social state structure, I don't know if public sector is compensating or incentivizing something fully. This is not something I can talk about, but up until now, the things I have heard tells that this would be a disadvantage.
Thank you, Professor Demirci. Is there any other question by the participants?
I see a hands up by Noura Birimo, but I don't know if it's a hands up for a question.
Yeah, I want to thank you again. Like my question is more general, but I always asking myself, like, as a researcher, every one of us like aiming to transfer the products we got from our researchers or our experiments. What the extent of society's acceptance of such type of devices, for example, as a startup or anything else like that?
I mean, I guess depends on what you envision your goal is with one's career. I kind of envisioned that what I built, at least one thing I built in my career, would impact somebody beyond my career. So that was my starting point when I started as a PhD student. I said, at some point, someday, I'll do something. I don't know when, what, how, but it will help other people. So for many years, I did HIV research for global health, developed things, and that was. And then one thing, coming back to your question is, the only way that a tool that you publish becomes available to others, that it has to go through this whole really challenging life cycle. And then it has to be tested pretty much with the fire of the market, fire of the clinic and clinicians, that it's truly needed. So when it goes through all these different exams, so to speak, if you think about it, beyond the first paper you published on it, then one day might be used for people and can lead to benefit of others. But I guess the only way to do it is to really translate it out of the lab, and that's I don't know, in our current age and time, that's the only way to really make it available to others. So I mean, I know people have used this device in South Africa, and they would have never used it unless I try to make it available or work with the right team. I mean, I'm still an academician. I'm doing my academic work, but I built a team that would take it and carry it forward and make it what it is. And,
so we can say like the case is the time, or if the situation is the same for cancer, let's say.
I didn't get the question.
Like, is the situation same for the cancer case, or like it's gonna take more time to approve our case?
Depends, depends on the tool, depends on if it's a drug, it's gonna take much longer, right? If it's a device that is a predicate, then it might be easier to get an FDA approval. It all depends on what the tool or what the solution is. I don't think there's a cookie cutter solution across different scientific developments. They're all very different, and they all have their very different challenges. You just have to be willing to, push it forward through these different challenges.
Yeah, I got it. Thanks a lot.
Thank you, Mrs. Primo. Thank you, Professor Demirci. I do not see other raised hands. I would like to mention that we need help of all the participants of this webinar to improve our future events, and we would like them to complete a 1-minute survey that we have placed the link in the chat. So please do not hesitate to use to click there and to fill in the forms. It will be very helpful for us and the future speakers. I think that we have come to the end of the webinar. I would like to thank, of course, the speakers, Professor Öztürk and Professor Demirci, the technical team for supporting the organization of the webinar, and all the participants who have come so many to listen to these talks. The next webinar will happen in September 2022. It might be transformed into a seminar and face-to-face directly at the Center of Life Sciences in Istanbul. So stay tuned for news about this upcoming webinar. Thank you very much, everyone. Thank you, everybody, and goodbye.
Thank you. Have a great day, everyone.
Thank you.